All right, hello. Welcome to the other segment of Chapter 8. We're going to take a look at this chemical reaction. So you should be familiar with chemical reactions in general, right? So we have a one reactant here. Okay, so remember these are the reactants. Okay, and then these are the products. We have two products, two reactants. Okay, and in the middle here, over the arrow where it's showing the direction in which the reaction is occurring. Now, it could go the other way as well. We have this word right here. Now here, the reactant is sucrose plus water. So we've got hydrolysis going on. So we've got the addition of water to break down sucrose into its two monomers, right? So it's a dimer, it's a disaccharide. Now we have monosaccharides here. We have glucose and fructose. Now, this may occur, right? Just hydrolysis may occur, but it could happen a very, very long time. It could be days, it could be hours, it could be minutes, it could be weeks. But we have this right here. This is an enzyme. It's called sucrase. Now, many enzymes are named based on their function or the molecule that they bind or break down or make, right? So it could be the molecule that they make. Now, this one happens to bind to, so what's going to happen is this reactant is going to combine with sucrase, the enzyme. And again, remember they're proteins. It's a protein. And what it does is catalyze the chemical reaction. Okay, catalyze. Okay, catalysis helps speed up a chemical reaction, so it makes it go faster. Okay, so in the presence of this enzyme, this chemical reaction will happen faster. It could be a fraction of a second. That's how well enzymes help or catalyze chemical reactions. All right, so let's further. We're going to look at this curve right here. Now, this shows the progress of the reaction. So we can imagine maybe it is the reaction that we just looked at. We have sucrose and water here, and then we have fructose and glucose here, and then we have the enzyme that could be helping catalyze it, but it may happen without it. So let's just look at a general progress of a reaction. So we have the progress, so as it goes from reactants to products, so we have reactants, products here. And then we have, remember, free energy, right? So this is the available energy. So remember the reactants start out at a particular energy, and then the products are going to be have a particular energy associated with them. And here we've got the reactants at a higher energy level than the products. So we can imagine that the starting free energy occurred here. The ending free energy occurred here. The free energy went down, so it's going to be negative, right? So this represents an exergonic reaction. Now I want to kind of talk about what happens in between there. So look, we've got this hill, and then we need to go up, and then we need to go down. Okay, so it actually eventually goes down. Now, this right here, the E sub A, is called the activation energy, or the energy of activation. So that's really the energy that must be kind of present in the system or be put into the system in order to get the reaction started. Okay, so we need the reactants, right, to start to do something. And what they're doing in the middle is this transition state, right? This is a transition state. So you can see the little squiggly lines. Okay, these squiggly lines are representing the breaking of these bonds, and then eventually new bonds will be made. Okay, so the transition state is very unstable. Okay, and this is where breaking bonds occurs and making new. Okay, and this all refers to bonds. Okay, bonds. Okay, so this represents basically what's happening during a chemical reaction in terms of energy. We're going to kind of go back and refer to this. So let's take a look at this curve. It's very similar to the one that I covered before with just the chemical reaction. However, this shows two curves, right? So there are two curves, and one of them represents the chemical reaction with the enzyme, and one of them represents the chemical reaction without the enzyme. So we can see here that the black curve represents without the enzyme, right? So we have without. Okay, and then we have a red curve, which represents with the enzyme. So you can see they have very similarities, but however, they do have some differences. So remember, this is the progress of the reaction, moving from reactants to products. This is the free energy, right? So the available energy to do this work. And the reactants, of course, are going to start out at a particular energy. So they have an energy associated with them. Now, for both curves, the reactants start out at the same energy because, of course, they're the same molecules, so they're going to have the same energy. And then the products have an energy associated with them. So this, again, represents an exergonic reaction because you can see that the reactants start out at a higher energy level than the products. So the free energy goes down. Right? So the free energy goes down. So this is going to be negative, right? So delta G is negative. Now, here it says also the delta G is not affected by the enzyme. Okay, so remember we have two curves here. These curves, one of them represents without the enzyme, one of them represents with the enzyme. So the change in free energy does not change, right? So the change in free energy is the same with the enzyme as it is without the enzyme. Okay, so delta G enzyme equals delta G no enzyme. Okay, now let's look at the curve. Okay, so we have a higher hill to overcome without the enzyme. Okay, so without the enzyme, there's a much higher hill than it is with the enzyme. So we can go ahead and just show this right here, even though it's to the right. So we have the activation energy with the enzyme, the activation energy without the enzyme. Okay, so you can see the activation energy without the enzyme is a lot greater than it is with the enzyme. So enzymes, as you can see, lower the activation energy. And that was described in the animation that you just saw. So without the enzyme, there's a greater activation energy that must be, basically this hill must be overcome. It must be, that hill must be reached before it can reach to products. If the hill is shorter, okay, then it takes a lot less energy. So think about when you're walking up a hill. If you're walking up a large mountain versus a hill, which one would take more energy to get to the top? Okay, it's the same idea. So enzymes lower the activation. So let's look at an enzyme. So here represents the space-filling model of an enzyme. And here we have what's called, this is a space right here, okay? It's the active site. The active site is where the substrate binds. So the substrate's going to get in there, okay? The substrate is typically shaped, right, and charged complementary to the active site. So it kind of fits right in there nicely. Now, there's not always only one substrate. There could be more than one substrate. However, this is just showing one substrate, and this substrate fits right into the active site. And it's the active site where all the stuff is happening, right? So this is going to be where the chemical reaction takes place. Now, once a substrate binds, it often induces a change in shape of the enzyme. Okay, so here we have now the enzyme substrate complex. So this is E plus S. Now we have ES. Okay, so we have the enzyme substrate complex. You can see the shape of the enzyme has changed. It's kind of closed, right? And it's almost like hugging the substrate. It's giving it a nice hard hug, right? So what is happening in there when it happens? There's a lot of things happening in there. Okay, so it's this that helps the substrates change to products. Okay, so here this is induced. It's called the induced fit model. Okay, so when the substrate binded, it induces, right, a change in shape of the enzyme. So it kind of causes the enzyme to kind of wrap around it. So
Watch
let's
how
look at
substrate binding
this
to an enzyme active site
animation.
induces a change in the tertiary structure
And
of the enzyme.
this kind of talks about
These
that
images were
same
created using X-ray
idea.
diffraction data of hexokinase with and without bound glucose. Notice how glucose binding to hexokinase causes a conformational change in the enzyme that closes the active site. Credit: Mark B. Gerstein, Yale University.
Okay, so that kind of shows its movement. This was actually done. This was an experiment. So this kind of. So let's talk about the catalytic cycle of an enzyme. So enzymes go through a cycle. And the cycle starts out with the enzyme and the substrate, and then it's going to end with the enzyme and the products, right? So it always starts and ends that way. All right, so let's look at how the enzyme kind of starts. So we've got the enzyme, plus we have the substrate, and remember you have that active site, right? So you have that active site, and here we've got two substrates. Both the substrates are binding. They fit in their perfect spot, right? So you can see they've got this spot right here where the shape of each of them kind of correspond to the enzyme lowers the activation energy so that basically it doesn't require as much energy to get the reaction. And then, of course, the products are going to form. Now, the products have a different shape than the substrates, and the enzyme is typically has more of an attraction to the substrates than it has the products. So basically, the enzyme typically just releases the products pretty quickly once they form. And then is basically the enzyme goes back to its original open shape, right? So it started out this way before the substrates bound. So this is the enzyme, which is available to do another reaction. So this is its activity. Okay, so the activity is the rate in which an enzyme converts products or reactants to products. Okay, so it's a rate. Some enzymes are allowed to go faster, some of them allow the reactions to go slower. Now, let's talk about how the enzyme actually lowers the activation energy. Okay, so remember the enzyme has this active site, and it's the active site that actually lowers activation energy. How does it do this, right? It orients the substrates correctly. So it puts the substrates in their correct order because they can only fit one way into the active site. And that way is the preferred way to get the chemical reaction going. It strains so it could be not all of these, but it could be one or more of these things can happen. So it can strain the substrate bonds. Okay, so it can strain those substrate bonds. How it does that is, remember that induced fit. So it kind of closes in on the substrates, and it kind of strains those bonds by like squishing the substrates together. And all of this really provides a favorable microenvironment, right? So this is favorable for the reaction to proceed to form products. And then sometimes the enzyme forms covalent bonds with the substrate. When it forms covalent bonds, bonds are broken, right? So that kind of contributes to the breaking of bonds, and then of course eventually making new bonds. Now, there are things that can like affect the activity of the enzyme. So remember, it's a protein, so it's pretty sensitive to its environment. So we're going to talk about some of those things. Okay, so let's take a look at this curve. Here it says, at the very top, it says effect of substrate and enzyme concentration. So we're going to kind of look at that. Now, when you study the activity of an enzyme, what you're going to do is basically set up chemical reactions where you have a certain amount or varying amounts of enzyme. But in one particular chemical reaction, you're going to have a particular enzyme concentration, and then over several chemical reactions, you're going to have substrate concentrations that increase. So here, this represents the activity of that enzyme as you increase the substrate concentration. So here we've got the increase in substrate concentration across the bottom, and then here the velocity. Okay, so here this is the initial velocity of a particular enzyme, but over time it's not really the initial velocity anymore. What that means is it's the rate in which the enzyme converts reactants to products. That's its activity, right? So I kind of just defined that earlier. So an activity of an enzyme is the rate in which it can convert the reactants to the products. So when you look at this, it starts out very fast, right? So it increases significantly. Okay, when the substrate, even with the substrate relatively low. But as the substrate increases, it starts to level off. So you would expect, so initially you can see that it increases as the substrate concentration increases, but then that rate starts to level off. Okay, this is where it becomes saturated. So the enzyme becomes saturated. So there's a lot more concentration, relatively speaking, than there is enzyme present. So the enzyme is basically overcome by this. This is called the maximum velocity, or Vmax. Now, if we look at this, we've got 1/2 the Vmax. So 1/2 the Vmax is really 1/2 the way up this, exponential rate right here of enzyme activity. Well, at 1/2 Vmax, if you kind of follow this line, straight line, all the way down to that particular substrate concentration, whatever that is, that represents the Km. This is the Michaelis constant. It's actually used to study enzymes. So if there's a small Km, that means the enzyme doesn't need a whole lot of the substrate to become saturated. If there's a larger Km, of course, that's just the opposite. So then it needs a lot, the enzyme needs a lot more substrate. Local conditions can affect the activity of the enzyme. So remember, the activity is the rate in which an enzyme converts the reactants to the products. Now, environmental factors and then chemicals that may or may not need to be present in the enzyme or as part of the enzyme may affect its activity, whether or not it's there. So let's look. In terms of environment, the temperature can matter. So we can see this is a figure from your textbook showing the rate of reaction. So this would be the velocity. Okay, versus temperature. So as we increase temperature, instead of changing substrate, right, each one of these have the same amount of substrate. It's just we tested different temperatures, or they tested different temperatures. Well, this compares two different enzymes. So certain enzymes prefer certain temperatures. So they work best, or they have their greatest activity at certain temperatures. So if we look at the human enzyme here, our normal temperature is about 37 degrees Fahrenheit, or Celsius. Okay, at 37 degrees Celsius, the enzymes in our body should be at their maximum potential, right? So they should be working and their activity should be at the maximum. So this kind of shows that, right? So at the peak of this curve, this represents the greatest activity at that particular temperature, at that particular temperature. So this happens to be, of course, 37 degrees. Then if we look at one from a thermophilic bacteria, those are organisms that live in very extreme environments. They're called extremophiles. So this one has its particular enzyme has its highest activity at about 75 degrees. Okay, so if you put the thermophilic bacterium in a human, it wouldn't have any activity, right? So our temperatures are about 37 degrees Celsius. Look, it's got 0 activity at 40 degrees Celsius. If we put a human enzyme inside the thermophilic bacteria, it also would not have any activity. Usually at low temperatures, the molecular movement, okay, so diffusion and the movement of the molecules are so slow that the activity is so low. At higher temperatures, or those temperatures that are too high for any particular enzyme, remember it's a protein, if the temperature is too high, it can denature, right? So when an enzyme denatures, it's not in its native state, so that it's not going to have a great activity, if any at all. In terms of pH, right? So the pH in the environment can affect the activity of the enzyme. So here we are comparing 2 human proteins. We have pepsin and trypsin. They're in different locations in the body. So pepsin is found in the stomach, trypsin is found in the intestines. Now, we can see that peptin's greatest activity, right? So its preferred pH is at 2, right? So that makes sense because it's in the stomach where it's very acidic. So it's going to work best in acidic conditions. And then we have trypsin. It's going to be found in the small intestine. It helps break down foods. And in the small intestine, right, so this particular enzyme, it's going to be more alkaline, right? So it's in the small intestine. So, and of course, its greatest activity is going to be in more alkaline conditions. And in this case, pH 8 is the preferred pH for trypsin. Now, like I mentioned, some chemicals, whether they're there or not, could affect the activity of the enzyme. So cofactors are actually not proteins, and they're different types of molecules. So there could be an organic molecule or inorganic. When they're inorganic, it's usually a metal ion, right? So it could be magnesium, it could be iron. Sodium could be an example of an inorganic cofactor. An organic cofactor is going to be something like that has carbon in it, so like a vitamin, right? So organic cofactors are actually called coenzymes. Okay, so that's a specific name given to cofactors that are actually organic. And some examples would be vitamin C. Vitamin C is kind of an important cofactor for certain enzymes in the body. Vitamin A could serve as a cofactor. And then we're going to see another cofactor when we move on to chapter 9 and 10. It's called NAD. Now, cofactors often are part of the enzyme. So they're required to bind the enzyme for the enzyme to have its activity. So here we've got kind of a representation of this. So here we have the enzyme without the cofactor. And the enzyme without the cofactor isn't going to bind the substrate. So the substrate's not going to fit in there correctly. But with the cofactor, so let's say we add the cofactor, now with the cofactor, the substrate fits nicely. Okay, now cofactors do a number of things, but it does help the enzyme or basically make the enzyme active. Now, there are other molecules that can inhibit the enzyme's activity. So it kind of works against it. So let's look at this enzyme right here. So we've got this purple blob. We've got this enzyme, and it's got a few kind of, indentions in it. Those are actually sites where molecules can bind. So this one represents the active site. And this represents the substrate. So this is a normal substrate for this particular enzyme. So you can see it kind of fits right in there nicely. We've got this little notch here. It fits right into that little indentation right there, right? So this is the enzyme in its normal shape and the substrate, right? So it fits perfectly. So that's normal binding. So that's what would happen in the normal case. Now, in the presence of a competitive inhibitor, okay, so a competitive inhibitor is just another chemical that can bind to the enzyme. Well, competitive inhibitors actually bind to the enzyme in the active site. So when they bind in the enzyme in the active site, it prevents the normal substrate from binding. So it basically blocks it. Now, a non-competitive inhibitor, okay, is a molecule that inhibits the enzyme's activity, similar to the competitive inhibitor, except it doesn't bind to the active site. It binds somewhere else. This is usually, see, you can see this other notch right here. This is called normally a regulatory site. Okay, so often regulatory sites bind to non-competitive inhibitors. So once the non-competitive inhibitor is present in the environment, it's going to bind to that regulatory site, and then it changes the shape of the enzyme. So when it changes the shape of the enzyme and the enzyme's active site, it keeps the substrate from binding normally. So then it lowers the activity of the enzyme. Now, it must be noted that competitive inhibitors can be overcome by changing the concentration of the normal substrate. So if we increased the substrate concentration, the normal substrate concentration, it's more likely that will bind. So that can compete for the active site with the competitive inhibitor. So they're both competing for the active site. The one that has the highest concentration wins. So let's talk about regulation of enzymes. They must be controlled. Sometimes you don't want the enzymes to work, right? So let's say we have a product, we have enough of that product, we don't want to make any more of that product. It causes chemical chaos in the cell, right? So it would just be mass chaos if all the enzymes were working at the same time. So they must be regulated. So there must be switches that turn them on and off. This is called allosteric regulation. Allosteric regulation can activate an enzyme or stimulate its activity, make it work faster, or it can inhibit the activity. It could slow it down or stop the enzyme. So we don't want chemical chaos. Let's take a look at allosteric activators and inhibitors when they kind of act upon an enzyme. So here, this is the normal enzyme without the activator or inhibitor present in the environment. So a lot of allosterically regulated proteins, they can alternate between an active and inactive form. So this shows the active form, which is kind of open, right? So you have the active sites, they appear that they're open and ready to bind an activator. And then we have the non-functional active sites kind of closed in the inactive form of the enzyme. However, it leaves this space open here. So let's look at what happens when an activator or an inhibitor is present in the environment. So when there's an activator present, it binds, and basically it fits right into that spot right there and stabilizes the active form. So basically keeps the active form active until the activator is not present anymore. So this could be produced by the cell, or it could be something that is introduced as a pharmaceutical of some sort. So this basically stabilizes the active form. So now the enzyme is doing its thing. So it keeps this open active site, right, open so that its natural substrate can bind. Now, if an inhibitor is present, it binds to the same spot. However, it fits just right in the inactive form. So it's going to bind to the inactive form, and it's going to stabilize the inactive form. So you can see the inactive form, the active site is not open. So the enzyme is not going to produce product, right? So it's not going to catalyze that chemical reaction. So this inhibits the enzyme. So this is all the same enzyme. Okay, it can be present in an active form or an inactive form. This is called allosteric regulation. Some of these types of enzymes, which have multiple, I guess, subunits, right? So the quaternary structure, they exhibit cooperativity. Actually, another form of allosteric regulation, which actually increases the activity of an enzyme. So what happens is we have the inactive form, and a substrate can increase in concentration enough where the substrate can bind one of the active sites. Okay, a key, I guess, model of this cooperativity is hemoglobin. Hemoglobin can bind 4 oxygens, but it binds it in such a way that it must bind 1 oxygen first, right, before the other 3 sites are ready to bind oxygen. Once that one binds, it opens up the other 3 sites so that the substrate can bind those 3 pretty quickly. So it's not gradual as we saw with the activity curve previously, and it shows a different shape, the curve, in terms of activity for these types of enzymes. Feedback inhibition is another way to regulate enzyme activity. So remember the small metabolic pathway that I kind of introduced in the beginning or in that other segment? Okay, so this represents a metabolic pathway. Now, with feedback inhibition, often the end product of a pathway serves as an inhibitor, right? So if the end product is at a high enough concentration, so basically enough of it has been produced, it can come back and bind to an enzyme early in the pathway, right? So let's say we have enough of this product. Now there's so much of it that it binds enzyme 1 in this pathway. Once it binds enzyme 1, it stops enzyme 1 so that A isn't converted to B, and then B cannot be converted to C, and then C cannot be converted to produce more D because we don't need any more D. Okay, this is feedback inhibition. Now let's see an actual. So we have this pathway right here. This is actually what happens in the cell. We have a substrate called threonine. Threonine is an amino acid, but threonine is converted to isoleucine, which is another amino acid inside the body, using this pathway here. Now, threonine binds to the active site, so this is the natural substrate of this enzyme threonine deaminase. This is enzyme 1 in this pathway. So then threonine deaminase converts threonine to an intermediate, and then enzyme 2 converts it to another intermediate, and then so on and so forth until finally isoleucine is produced. All right, so now we have some threonine, and we have threonine being converted to isoleucine. So now we have isoleucine present in the cell, which is good because isoleucine is needed for many proteins. Now, let's say this pathway keeps going. We still keep getting threonine, and we are making more isoleucine, but we don't need any more isoleucine. Let's say isoleucine increases in such a high concentration that it serves as an inhibitor, right? So isoleucine, at a high enough concentration, it's going to bind to this regulatory site in threonine deaminase. When it binds to the regulatory site in threonine deaminase, it stops that enzyme. So it could basically block the enzyme. So it changes the enzyme's active site so that it's no longer going to bind threonine. Now, let's say this continues to happen. Now we've stopped this pathway, so now we're not making any more isoleucine. So now isoleucine is going to start to decrease in concentration, and then it's going to be used up by the cell to produce proteins. So then it's no longer going to be present in a high enough concentration to be able to bind to this enzyme. So when it doesn't bind to the enzyme is active again to produce more isoleucine. So you can see it's a cycle, right? You produce enough isoleucine, it stops the pathway because we don't need any more. But then when the isoleucine starts to get used up by the cell, it automatically is restarted again because it's not present to bind this enzyme to stop the pathway. Pretty interesting, huh? A lot of enzymes work this way or are regulated in this way. It's called feedback inhibition. All right, so I want to kind of, one final note before we move on to Chapter 9, and this kind of is going to become important in Chapter 9. Enzymes are localized where they're needed in the cell. Okay, so they could be transported to the location that they're needed. But sometimes they're made in the location that they're needed. So an example would be in the mitochondrion. So remember the mitochondrion, it's thought to have used to been a bacterium. So it has its own components to be able to produce proteins. Okay, so it has ribosomes in there. There's DNA in there. It has its own DNA. So it can produce proteins. And usually the proteins that are involved in cellular respiration, which is where this happens, right? So cellular respiration occurs in the mitochondria. So of course the proteins that are required for cellular respiration, it makes sense that they are made there. Okay, so here we've got the matrix, which is that semi fluid substance, right? That's inside the inner membrane of the mitochondrion. There's enzymes in that fluid, which are actually part of the Krebs cycle, which is the second major stage of cellular respiration. And then there's a third stage of respiration, or the last stage of respiration, which has proteins that which is actually occurs in the inner membrane in those cristae, right? So them foldings right there in the inner membrane. So the proteins are embedded in the inner membrane, of course. Okay, and most all of them are made in the mitochondrion, so it makes it a lot more efficient. All right, so we're going to be kind of revisiting these structures right here. So the parts of the mitochondrion and the chloroplast for chapter 10, and of course 9 for the mitochondrion and cellular respiration. I kind of talked about enzymes. I talked about their function and how they work. How do they lower the activation energy? Talked about the conditions that affect the enzymes, and to talk about how enzymes are actually regulated. All right, so I think we're ready for chapter 9.